Okay, we're at 9:01. I think we still have some more people joining, but we'll go ahead and start for everyone's time's sake. So welcome to, good morning, good evening, or good afternoon to those, whoever you are in the world. Welcome to the Emerging Research Showcase today. We have an exciting lineup of presenters, and I hope everyone will be able to enjoy it and make some connections. I do want to express our appreciation to Alexandria Realty for sponsoring today's Emerging Research Showcase. And with that, I will turn it over to Steve Lommel.
Thank you, Adrienne. First of all, welcome everybody. I wish we could meet in person. I see a lot of names and faces here that I haven't seen in a long time, so it's at least good to see you virtually. And then hopefully in the not too distant future, we can meet again in person. This is a timely showcase international programs. NC State University is very serious about advancing our international programs. We are in the middle of a strategic planning process at the university level in which international programs has become much more front and center. And most of the colleges follow in the slipstream of a university's strategic plan and do their own college's strategic plan to align with the universities. And we too will be working on one in the college and featuring international program activities much more. The reason for this, it makes for a holistic institution. It allows students to have a broader experience, it enhances research, and it's really obvious that it would impact our university, the quality of the science and the quality of our graduates, and also probably improve our rankings as well. So this is something that we are thinking about currently, and it's really in our consciousness. So this meeting today will be informative to me to hear and learn about opportunities and where we can make some key investments to advance our international program activities. We've had a long tradition in international programs, but when we compare ourselves to other institutions, it appears that our level of activity may not be as high because we have yet to achieve some of what I would call the gold standards or hallmarks of major international program activities, and some of these include some of these major USAID type of grants, but we're working on those as well. I'll pretty much stop there, but I just want to thank all the speakers. I looked over the session today, and it's very exciting, and I'm going to stay online and listen to this because, again, international programs is front and center for the college and the university, and it will help achieve our goals with the Plant Sciences Initiative, with the Food Animal Initiative, and many other of the big initiatives we have in the college. So I'll stop there, Adrienne, and once again, thank everybody, and looking forward to a great morning of great ideas and talks.
Thank you. Now we'll turn the floor over to Erin Meyer.
Hello, good morning. It's good to see a great turnout this morning for this important conversation. So thank you for being here. I'd like to echo Adrienne's thanks for Alexandria Realty being the sponsor today. We really appreciate their support of these emerging research showcases. One of my roles here at NC State is working with the CALS Research Foundation Board, and it's great to see a couple of you on this call. Thank you for your participation. And I'm here this morning to let you know that if you're considering how you might invest in or support faculty research initiatives, I'd ask that you talk to me about supporting the CALS Research Innovation Endowment Fund. It's a fund that the Research Foundation Board of Directors annually awards funds to faculty through a competitive process. And we have evidence that these awards have helped past recipients in attracting additional funding, as well as receive national and international recognition for the college and the university. So to the companies and individuals that are on this call that have supported us already, thank you very much. And for those of you who haven't yet, I hope you'll consider supporting us so that you'll see more of what you can see a little bit more of what you'll see today. So thank you, and I'll turn it back over to Adrienne.
Thanks, Erin. Now we'll turn it over to Jose Cisneros.
Good morning, and thank you for being here this morning. For the ones that do know me, I'm Jose Cisneros. I'm the Director of International Programs for the College of Agriculture and Life Sciences here at NC State. Before we go with the program that I promise will be an excellent program, we have a great panel, and then we have a selected group of scientists that will present their international research. I would like to talk about 4 points that I believe are why it is important to have international recognition. When you have international reputation, the best international students will look to come here to study. When they go back to their home, they will be the leaders of the country. This will mean lifelong strong relationships with us. That will mean more business with the US and more business with North Carolina in particular. When you have international reputation, you will be consulted. That means you will be sitting in the table with policy and decision makers. When you have international reputation, scientific companies will invest close to you. More funding for NC State and investment for North Carolina. Finally, when you have international reputation, funding organizations will be investing in our research. So this is a good investment. I have seen this before. I have been in international for 15 years in 3 different universities, and the investment in international pays off in the long run in a lot of different ways. So if you have any interest, please contact me, Erin, or Deborah Thompson. Thank you, and please enjoy this event.
Thank you. We'll have our panel discussion next. Before I hand it over to Ryan Combs, our moderator just has some logistics. See, everyone is muted, so well done there. I think we're used to Zoom land now. This is being recorded, and so everyone who registered for the event will receive a link to the recording at the end. In the next week or so, Deborah Thompson, the Director of Research Partnerships, who I'm standing in for right now, will follow up with each person each organization is attending with any contacts to make any further action. If you have any interest in connecting with the PIs, please feel free to do so. The next Emerging Research Showcase will be happening in the fall. If you have any suggestions about themes, please share those with Deborah Thompson. And as a reminder, no confidential information will be disclosed during this event. And at the end of the sessions, we will have some breakout rooms for you to connect directly with the PIs. So if you want to stick around for that, we can, you have the option to connect with the different PIs with the breakout rooms. So now I will turn it over to Ryan Combs, who will lead the panel discussion.
Well, Adrienne, thanks so much for the introduction. And as Adrienne mentioned, my name's Ryan Combs. I'm the Executive Director of the Research Triangle Partnership. We're a 12-county regional economic development organization, and our mission is really to go out and market the Triangle externally. We found that North Carolina is still not very well known internationally, so we spend a lot of our efforts trying to build our brand globally, specifically in Europe. And we've had incredible opportunities to meet with and connect CALS to both companies and research universities in Europe. I also sit on the CALS Research Foundation Board, which gives me an opportunity to better understand just how incredible the research is that's being conducted here. I'm very proud of the fact that in 2019, NC State received more USDA funding than any other university in the country. And I think it's a testament to our faculty and our researchers, and it proves that we're becoming the go-to university for agricultural research. As Dean Lommel mentioned earlier, with the PSI coming online in February of next year, we're now positioned to become the desired location for international science. We have all the advantages needed. We just need to make some strategic investments to help us build our brand and help us gain the notoriety that we deserve. Today's panel can help us better understand why international collaboration is so important in terms of driving international reputation. So at this time, I'd like to bring into the conversation each of our distinguished panelists, and I'll introduce those now. We have from NC State, Mario Ferruzzi, who's a Ph.D. professor for Plants for Human Health Institute in the Department of Food, Bioprocessing, and Nutrition Sciences. We also have Tomohisa Koyama, who's an associate trustee and an advisor to the president and executive director for NU Tech, the technology partnership of Nagoya University of Japan. We also have with us Paula Barngrover, the CEO of Iluma, which is a Colombian company here in the Triangle. And then finally, we have Jim Lorenzen, who's a senior program officer with the Bill and Melinda Gates Foundation. Last week, I was able to have fantastic conversations with each of our panelists. And one of the best quotes that I took away from those conversations came from Mario, who said, part of building a reputation is being present. Investments come after reputations are built. And I thought that quote was so relevant for today's conversation. So just keep that in mind as we talk. Part of building a reputation is being present. Investments come after reputations are built. So for today, I've prepared some questions to just start the conversation, but by all means, if anyone on the call has questions, I want you to feel free to interject. And if you want to put questions in the chat screen, just let us know. But I wanted to start with Jim, if he's on the call. I just want to make sure he's on because I know our time is limited with Jim. Jim, are you here?
I'm not seeing him as an attendee. Okay.
No problem. So I'll kick it over to Mario. You know, as I said, we had conversations last week with all the panelists. And I loved my conversation with Mario. Mario and I discussed the fact that Mario spent time both on the academic side of things as well as the private sector, working for conglomerates like Nestle, doing international product development in places like Africa. So Mario, can you talk to us a little bit about how the connections that you made working internationally have helped you in your current role at NC State?
Sure, thanks, Ryan. So to give everyone just a quick background, I'm a regular faculty member that has a 100% research appointment to engage in international research with partners in Europe and as well as international development with projects funded primarily right now through USAID. My experience started very early in terms of international. I think it was partially the way I grew up, being in a family that my father traveled internationally and being an international here in North Carolina, growing up in North Carolina, but being born from Italy. What was really interesting to me, though, is I always had an eye on kind of getting the field view of what's happening in the area of food and nutrition, which is what I work in. And my first experience with Nestle allowed me to kind of broaden the views that I was getting from graduate school and research and begin to transition them into what a global, what I would say, perspective of the food sector, of the nutrition sector, and by extension, the ag sector is. So it taught me a lot of perspective. And it also taught me that, I think like you mentioned, Ryan, the quote of being present, it taught me that, you know, in this day and age of COVID where everything is done by Zoom, how important it is that we are physically at these sites and locations when we're working. This is particularly important in a lot of the development work that we do with USAID through the Food Processing Innovation Lab and the Sorghum and Millet Innovation Lab. Activities go on without us, but being present puts you in the room to anticipate what the core opportunities to really have impact are. And I think I got that from doing international nutrition type research as a grad student to moving to a multinational and then growing as a faculty member through Purdue, where international programs was highly, I would say is a big part of the effort in the college, but also the university. And it was kind of ingrained in my culture as a young faculty member that there's always a place for that if you want to do it. And if you do, we're going to invest and facilitate that, because we know that when you're traveling around and you're present, you're carrying that brand along and making people aware of where good work is done. And then that brings the next step. So that's just the start, but little bit about what we talked about last week.
Yeah, absolutely. Tomohisa, let me kick it over to you. So Nagoya has partnerships throughout the world, including Germany, Thailand, and Shanghai. Tell me why global partnerships are important for your university.
なんかこれどうなってんの？岡田さん、これオンラインになってる？俺なんかフリーズしちゃったんだけど。
There we go. All right. Well, let's kick it over to Paula then, and we'll come back if he comes back to us. Paula, tell us a little bit about you and I talked. Your company spent a lot of time in Colombia before you decided to expand. Oh, are you back?
Sorry, I'm confused. So it's a freeze.
That's
Go
okay.
ahead. Go ahead.
Go ahead. We were asking why global global partnerships are important for Nagoya.
Yeah, Nagoya University, like any other industries, higher education in Japan, all over the world, competing globally more than 20, 30 years. And to attract good students and to attract good researchers, we need global reputations. And especially good researcher collect a good research fund. That is most important to us. That's why we need global respect.
Okay, thank you. And then Paul, I'll jump back to you. You represent a 40-year-old family-owned company that's based in Colombia. Until recently, your company was doing all their research internally. Just a few years ago, I believe about 5 years ago, you decided to look for international collaboration partners. Can you tell us why the decision was made to look internationally?
Sure. Iluma Alliance, for those who doesn't know, we focus on nourishing wellness, and we have different divisions for animal nutrition, animal health, human nutrition, and data science. So up until, like Ryan says, up until 2015, we were doing all our development and innovation internally. The company has always been a knowledge generating company. And then when we were analyzing the challenges that we were going to face as an industry, by understanding that the consumption will increase and the population will increase, and therefore the animal protein production, which is our focus, will need to increase 50%, we knew that we were not going to be able to achieve those increments in production by ourselves. So that's how we started looking to spaces where we could collaborate, so we could speed up our process.
Great, thank you. Jim, I see you've joined us. Welcome. I want to kick it over to you. I had a question prepared for you. You've spent most of your career working in international locations. You and I had a great conversation last week. I loved hearing your perspective on the fact that international reputation starts with personal relationships developed in the field. I think a perfect example of that is your relationship with Craig Yencho at NC State. Can you elaborate on that, on our discussion? Because I think it's such a valid point for us here today for our topic of discussion. Jim, you're on mute. There you go.
Sorry.
That's okay.
Yeah, thanks for the question, and sorry I joined late. I was thinking that the panel started a few minutes later than it did. Yeah, I've known Craig for a long time. We knew each other as potato researchers in the early 90s and kept in touch through the years a little bit. You know, he's a well-respected and successful sweet potato and potato breeder. And he's got a demonstrated commitment to education of African breeders. If you look at the country of Uganda, for example, he's been the Ph.D. supervisor of 3 generations, if you will. I mean, short generations of breeders there. And I used to be a banana breeder in Uganda, and we used to meet, you know, once or twice a year when he'd come to follow up with ex-grad students on the same research campus. And in addition to that, we held a convening to look at what were the options to develop genomic tools for sweet potato in order to accelerate sweet potato breeding. And Craig was one of the people that we invited to come to that as one of the leaders. And he definitely rose to be a clear leader among that group and the logical choice to lead an investment that we were making in genomic tools to accelerate sweet potato breeding. So, you know, a long history, but also an excellent reputation and technical expertise. So we were really happy to have Craig be that leader.
Great. Let me stay with you, and then Mario, I'd bring you in as well on the next question. You know, one thing that I didn't understand until our conversations last week, you know, Africa, the continent of Africa kept coming up. And I think, Mario, you can answer the part of, you know, why working in Africa can end up connecting university to global universities. But going back to Jim, you know, talk to us about why you believe Africa is the place to make connections, and why there's so many opportunities on the African continent.
Thanks, Ryan. Yeah, I mean, when it comes to modernizing agriculture and developing new markets, Africa is definitely the last frontier. The big ag tech companies are regularly, you know, dipping their toes in the waters, but it's still a high-risk market. But, you know, with its fast-growing population, Africa's the continent that will be requiring big increases in food production in the coming decades. Additionally, increasing food security and improving education in Africa are just huge opportunities to stabilize our future world. Poverty breeds insecurity, as well as the opportunity for new disease epidemics. And people don't realize, but around the world, economic transformation almost always begins with intensifying agriculture. Intensification requires improved supply chains, local suppliers, ag manufacturers, but it also generates ag surpluses that lead to new processing industries and the need for value chains. And it enables those families to have a cash income to keep their kids in school longer. So this all generates employment and skilled labor opportunities and leads to a virtuous cycle of growth in new industries and employing skilled labor. So Africa is a place to go.
Mario, you want to add in to that?
Yeah, I completely agree with Jim. And I think to build off the intensification of agriculture, a lot of our work is focused on building those value chains and making sure that they're robust, not just in urban areas where you have investments potentially from multinationals, where that relationship gets built, but also to solidify and build food security in rural areas. And so creating an infrastructure that allows for entrepreneurship, that allows for incubation of technology and transfer of technology is exciting in Africa because things can move very fast paced. We always have this notion of things move slowly in some of those parts of the world, but in fact, partnering with the right people, building those connections, you can make rapid progress in certain areas by starting from the ag infrastructure and then connecting it quickly to the value chain, which has real impact to people. So for us, you know, in our program, that is a fundamentally funded program from USDA and NIH and others. What we get from Africa is that diversity of thought, diversity of really enthusiasm for certain things that we help there, and it brings back ideas for us to deal with our specific research questions here. It's become a nexus of big thinkers as well of where to solve problems. And so it's important for us as researchers, I think, to think about what we can learn from our experiences in working in projects in international development that aren't just building reputation, but building the next set of questions and ideas and targets. So for me, it's great, and I agree with Jim. It starts from that fundamental focus on ag.
You know, and that was what was kind of enlightening to me is, as I mentioned earlier, you know, we spend a lot of time in Europe, and you look at the amounts of money of research dollars, especially in the EU for these academic institutions. And when you go meet with them, they talk about existing relationships they already have with UC Davis or Purdue or some of these other universities, but that's what kind of enlightened me on our conversations. Was it wasn't the relationship wasn't established there. It was probably established in Africa, working on a joint project together. So that's been a really eye-opening experience for me, just talking to you, talking to our panelists over the last week. Paula, let me jump back to you, talking about kind of building a global reputation. When you were making your decision on where to go in the U.S., I know your company looked at California, Florida, Georgia, Texas, and Massachusetts. What were the deciding factors that helped your company make the decision that the RTP and NC State was where you needed to be?
First of all, when we did the analysis, we didn't only analyze the United States, but other countries. We definitely wanted to locate ourselves in a developed country, and the United States was for us the one that made more sense. But then the question was, where in the United States? This is a large country. So we analyzed all those ecosystems, which from the animal production perspective, they all have advantages. But in our opinion, here in North Carolina, we found a special hub where we mix within the RTP all the sciences that we're interested in. We can count on animal nutrition, animal health, human nutrition, and better than that is that in order for us to be able to develop the technologies that we're interested in, as I said before, we're not going to be able to do it ourselves, and collaboration for us is a big part of it. And we analyze in order, like Mario said, in order for collaboration to be real, you need to be present. And we needed to decide where are we going to be present, so those informal collaborations, which for us are more powerful than the formal ones, can take place. And also the fact that I know you know, but 65% of students that go here in North Carolina, they want to stay in North Carolina, and talent is a great source for us to have, and the hardest thing to get. So we wanted to locate here where it is.
Great. And then Tomohisa, let me bring you back in. Your office has actually been here for 35 years. But one of the things you told me when we spoke was a lot of people back home in Japan don't quite understand this region, why you would choose to locate here for your U.S. office. And quite frankly, you said a lot of people have never even heard of us. So what do you tell those people back in Japan when they question why the triangle? I think you're muted.
I'm sorry about that.
That's
and
okay.
Actually, as our office established in 2007, but our connection with NC State University started in 1985. That means more than 35 years. And honestly speaking, people in Japan, normal people do not know Research Triangle Park or North Carolina. So I always say two things. One is biotic clusters. Research Triangle Park is one of the biggest biotic clusters in the United States. And number two is that there are good universities, not good, but great universities, NC State, UNC Chapel Hill, Duke universities. Some people do know these universities, so we always try to explain those two things and let people know, Japanese people know Research Triangle Park and North Carolina University and North Carolina. That is a win-win for us.
Well, and elaborate. So you not only partner with NC State, but you also partner with, is it Duke University as well?
Yes, because our university has a history of collaboration with Duke universities and medical schools. Our medical school has a student exchange agreement for a long time. Actually, these days it's getting inactive, but we have some connection with Duke and also Chapel Hill. But the most important university is NC State University.
Of course. Paula, let me go back to you real quick, because you also have partnerships with UNC and with Duke as well, correct?
Yes, correct.
Okay, can you elaborate a little bit on your partnerships with both Duke and UNC?
Yes, in Duke University, we have found collaborations in order for us to accomplish our goal of health through nutrition, and how to understand how nutrition can improve health itself without a medication. And with UNC, we work around the product development for human nutrition, and specifically the research campus in Canapolis is a great tool for us because all the capabilities that are there for, in my opinion, that ecosystem puts together not only the three big universities in the triangle, but also the other 7 that are in North Carolina, all contributing to nutrition.
Okay, thank you. Let me shift kind of gears a little bit, and I'll kick it back to Jim. You know, we talk about global reputation. I brought up UC Davis, and there were several other universities. Cornell popped up on numerous conversations last week, as did Purdue. Jim, when you and I spoke last week, you mentioned UC Davis made building a global reputation a priority. Can you elaborate on how they went about achieving that?
You know, Ryan, I'm really not an expert on the history of UC Davis, but I understand that in addition to a commitment to encourage faculty to recruit internationally, there was a parallel commitment that they made to assign personnel to build an international program staff, to build those relationships with institutions abroad. And as you know, it takes people, it takes time. Hard to imagine these days when travel's tough, but traveling and face-to-face relationships in addition to the long-distance relationships. And over time, those students who come and then go home and become leaders in their own countries become conduits as well. So it really is a long-term process. And I assume that's sort of what happened at UC Davis, as well as others in that highly ranked category that you mentioned.
Well, I'll kick it over to Mario. I mean, you went to grad school in the Midwest. You spent 12 years at Purdue University. When you were at Purdue, you told me they really began concentrating on international development. Can you talk a little bit about being a part of that discussion and process? I mean, why was it important to Purdue?
Well, so I mean, I think the way I would frame it is, I mean, Purdue has a long history of international development and commitment to it. And it ebbs and flows, right? So universities will go through transitions where they focus on different aspects of international, whether they're strategic partnerships with universities in Southeast Asia, China, Europe, et cetera, Latin America, or true international development efforts. It'll ebb and flow depending on the field at the time. When I got to Purdue, it was really a time when they were doubling down on certain efforts, and there was a transition in USAID to what I would say is the innovation lab model. And from that perspective, Purdue's long history in these CRISPs that they used to have served it to kind of move into that, continue those actions. As Jim said, there's a strong commitment to international programs, both at the college level and at the university level. And from that perspective, it trickles down in all aspects of faculty, from promotion and tenure to resource allocation, in terms of institutional commitment to grants that make the university more competitive when it's time to go about developing proposals and programs. But let's not forget one thing. It goes back to what Jim said. Davis has done, Cornell has done, Purdue has done, I think NC State has done as well, which is recruiting top tier talent from around the world. You start with that too, bring them in to bring some diversity of thought, some great leadership qualities from different parts of the world that actually feed into those programs and actually enrich them. It's very difficult to just home grow everything, right? So that's part of going around the world and developing collaborations, is identifying the talent that you can then bring in to further develop and become the next generation of leaders at your university as well. And again, Purdue has a long history of that. Ohio State in a different way. And NC State has a historical connection to a lot of the programs that we talk about as well, and an opportunity to grow in those areas. So I think the future is there, right? Just thinking about what we want, how to approach it. But the commitment has to be there from the individuals, people like me that decide they don't just want to play with beakers and test tubes and animal models in a lab, and commit to doing things in a translational way in a different part of the world. And oh, by the way, the benefits you get from that aren't just financial resources from the federal government or the Gates Foundations. It enriches every part of your program, and the reputation comes with it for the university, for you, et cetera. And it's a great thing to do if you are committed to doing it.
Great. Tomohisa, staying on that topic, you and I talked about why global reputation is so important for a university survival. Can you talk to the group a little bit about what we discussed last week on that?
You mean that why we need to survive in the global?
Well, just why it's so important for a global reputation in terms of, being able for universities to continue to grow.
Yes, as I mentioned earlier, we need global reputations because we attract the talented students and the researchers, especially in Japanese market. The high school student population is shrinking, and there are so many universities. And less students, that means that universities' quality is getting worse. And less students are going to graduate schools, and less students going to PhD programs. That is about our reputation of the research capabilities. So we need talented students all over the world. But unfortunately, honestly speaking, we collect intern students only from East Asia, and US students come to Japan, not only but to other universities. Some students, postdoc, come from United States to Japan, but that's not enough. So we need more students, and also we need more talented researchers. If we can collect the research monies, and if we can raise our reputations. In that sense, we can survive in the global market as a higher educations. That is our target.
Okay. Paula, when you were in Columbia, did you know about NC State? I mean, were they known to your folks, or talk to us a little bit about kind of their NC States, where they fit on the ranking of notoriety globally, and yeah, just elaborate on that if you don't mind.
Our expertise has been animal health and animal nutrition. So yes, we did know about NCSU back in Colombia and Latin America. The university does a good effort participating and publishing and participating in the United States associations like poultry science, animal science. So from that perspective, we do know the NCSU. In the international events, though, we didn't get to see them that much.
Okay.
And when we were talking about the importance of the global collaboration for the university, I don't know if you want me to go into why establishing the international cooperation office with more efforts will be maybe important for the university.
Yeah, please go ahead.
When we are seeing the international efforts of the university, of course, from the point of view of finding spaces internationally for the students is a good thing, but building up that global reputation that Mario and Mr. Tomohisa say is very important. That will bring, in my opinion, to the university the opportunity of accessing to global resources. When you do projects with international parties, then the research projects collaboration resources are higher, and not only from national sources. The international fundraising is an opportunity there, because as long as you build the reputation, then of course it's better to show the international efforts so you can find the international fundraising. And the international students, not only as going out from the United States to the world, but also allowing that interchange with other students, that always brings up better results, because when you join together the different capabilities of the collaborators, then you can speed up the process better.
Mario, do you want to jump in on this conversation?
Yeah, I will tell you, I agree 100% with Paula. I lived it in a lot of ways that so very much changed the way I think about recruiting students and postdocs now. In my years of transitioning from Purdue to NC State over 5 years ago, my laboratory was kind of built off of graduate students from 6 different countries, including the US, of course, as one of those 6, but Malawi, Ethiopia, Senegal, we had Ecuador and Colombia as well, and then the United States. And what was amazing was how they each approached things so differently. And in doing so, especially for the domestic students, whether they were the undergraduates or other graduate students in my lab, whether they were from Indiana or wherever, it didn't matter, or North Carolina, they're getting an international experience every day when they come into the lab. They're getting an international perspective on why someone else's project is important to them and their communities elsewhere. What we're doing to enrich the environment of our trainees and students and prepare them for a global workforce is incredible. You can't do that if you're singly minded and only training in your environment, i.e. your own students. That being said, we understand we're a state university, and our focus is, of course, on our stakeholders in the state of North Carolina. But this is where graduate education, research, extension activities, and translational opportunities can be enriched with that international component. And it can be kind of highlighted as a strength for what we do at home in North Carolina, why we're getting perspective even without leaving our offices, right? And it just creates a very different dynamic. And as a funny joke, you know, if you come to Kannapolis, the 4th floor of the Plants for Human Health building, 3rd and 4th floor, there's 3 of us that are natural born Italians that reside as one of the 12 faculty in the Institute. And the running joke is there was more Italian being spoken than any other language. So we realized we needed more diversity and started hiring, you know, American graduate students and others, because it's just interesting that you can find that kind of feel in a small town like Kannapolis, but it's a focus on us recruiting international talent. They don't count me as international, but in essence, that's a big point. And I just agree so much with what Paula just said.
Okay. Well, let me ask a question. This is kind of a group question, and anybody can kick in first. But, you know, NC State being a land grant university, I think a lot of times when people outside the university hear that we're globally engaged, it's kind of a negative thing. You know, it's what can international development, things we're doing internationally, actually help North Carolina farmers. So when someone brings that kind of question up, Mario, if you want to start, you can, and then we'll see what everybody else has to say. But how does what we're doing internationally help the farmers in the field in North Carolina?
So I think I've heard it best from a World Food Prize winner, Gabisa Jetta, who's here at Purdue. And I say that because I'm actually at Purdue this week working on a USAID project. So sorghum breeding and seed systems and things like this. And he said, you know, the land grant model that was signed into law, you know, Abraham Lincoln, everybody knows, the land grant system that the US established so in a visionary way is the ideal system to think about when we translate to international. And when we think about putting boots on the ground and actually having the trains run on time and actually taking research and putting it into practice. And why that's important is exactly what we were talking about. You broaden the notoriety of your university by engaging in these activities, which makes you a better place to recruit top-notch talent to your state to address issues globally but also be there and be present when your state needs something. And ideas that are generated from research that funds international research is bringing solutions to problems that are right at home. And whether it's generating novel breeding programs for a grain like sorghum that, by the way, the United States is the #1 producer of sorghum in the world, right? #1 exporter of sorghum in the world. But we don't consume it as much for human food here in the US. Gluten-free has changed some of that, but we do research for human food and animal consumption and use in Africa, but that research enriches our ability in our domestic businesses here for their export markets. And so to be able to have the genetic, the germplasm and everything else and be able to keep that here puts us at the top of the list. If anybody in the world, but more importantly, if there are any problems domestically that we have to deal with and overcome. So those investments just go a long way, besides the most logical one, which is recruiting workforce for your state that's the top workforce in the world. That's what you want. And so I want the best people from everywhere to want to come to North Carolina. It pains me when I hear that people don't know where we're at, or they think we're somewhere else, you know, than in the Midwest. So it's important in both of those paradigms for me, people and then solutions that you can apply locally.
Great.
Can I jump in?
Please.
Additional to what Mario is saying, in my opinion, yes, the university is very strong. The fact that it's a state university funded, and of course the farmers should benefit from it directly. And international collaboration will maintain that. Because the problems that we have here in North Carolina are not only from North Carolina, we find them somewhere else. And we are able to, through those collaborations, to find where is more economically efficient to solve those. And also the products that we manufacture or produce are not only consumed, like Mario says, in the United States, or not even within North Carolina. They go internationally. So it's important for those for the farmers to be able to access to collaborations and be able to solve issues and maybe drive innovations also in their destination of their products places.
Yes, go ahead, Tomohisa. You're muted.
I totally agree with Mario and Paula. My small experience is I'm working also as a vice president of Japanese Triangle Business Associations, that business association with 30 Japanese businesses. One big trading company's guys mentioned me. He tried to export chicken from North Carolina to Japan, but chicken's quality is very good in North Carolina. But he mentioned that no one knows North Carolina, and to put those ideas on the table, he had a very difficult time. If there is some world repetitions or the people know North Carolina and Sioux State University, that might be much more easy. He mentioned me to do. So in that sense, maybe building world reputations also helps local people to sell their product to globally. That sense in business-wise, I think definitely is building the world reputations helps local farmers.
Jim, do you have anything to add? I don't know if you dropped off, but the question was, you know, how does the work being done internationally directly help farmers in the field in North Carolina?
Yeah, I agree with everything that's been said so far by Paula and Mario and Tomohisa. But you know, one of the other things that's really important is, okay, you know, farmers know that they need markets, and you know, growing markets is important. And when you create more wealth in other places, that does create opportunities. But I'd rather focus on innovation and where innovation comes from. And innovation often comes from knowing what's possible. And as Paula mentioned, problems are common around the world. And so when you get more people together and you find out what's happening in other places. And sometimes in the case of working with Africa, you're really getting back to basics to see what are the foundations of what you do. You understand issues better. And then by bringing in global experts, and that's something we try to do, it opens up a whole realm of possibilities, and it sort of expands the bounds of where people think and where they look for solutions, because they're seeing things from different perspectives, different cultures. And so it really opens the doors for innovation and advancement, which is exactly where the place where universities need to be. And earlier, Mario had referred to the richness of having a culture where there are people from different universities, different countries, different places. And so it's improving the quality of education. It's improving the technical skills. I mean, if you look at what Craig Yensho is doing in his sweet potato and potato breeding programs, I would say it's radically different than it was when we started cooperation 6 or 8 years ago. And so he's moving to the forefront. He's bringing in top talent, and he's become a more efficient breeder using not only genomic tools, but different experimental designs, more efficient ways of analyzing, better ways to do more with less. And basically, he's going to serve the people of North Carolina better than he would have had he not had those connections. So I think there's many ways, quality of education, level of innovation and research, as well as the new markets that are developing by working in a global framework.
Okay. Well, let's kind of shift here and talk about what everybody thinks we can do, or North Carolina State can do to become better known and build our international brand. But, Mario, you and I, when we talked last week, you mentioned that any international effort has to focus on both big business and big institutes, but it also has to focus on the developing world. Talk to us a little bit about your thoughts on why this is so important.
Yeah, I think it went down the route of maybe building off what Jim just talked about, is creating that environment of innovation. And it's sort of like if you narrowly focus on one area or one group, if you focus primarily on Europe or focus primarily on Southeast Asia, you're not going to necessarily be playing in a truly global place, even though you're doing international work. And so from my perspective, I think it's important that from an institutional strategy, we think about international activities in truly global ways. And that means that some of it is, as Jim said, some of the times we're talking about like nuts and bolts in developing world. It's not high technology, but it's really application and translation of things that maybe we already do or that we can adapt very quickly. And that allows a level of engagement and participation in a playing field that also includes a lot of other players from around the world. But then engaging what I would say are the high-tech companies, and in my world, the food industry, is critically important, not only because it can help us drive really innovative high-tech research and lead to new projects and new collaborations, but they have a vested interest in the developing world. As Jim's talking about earlier, this is where large growth is going to have to occur if we're going to feed and improve the quality of life of millions and millions of people. And so when you think about it, in conversations that we have in developing proposals or projects, or moving projects along in our development work in Africa, my collaborators here at Purdue, Bruce Hamaker and others, we're always engaging the Nestle's of the world, the M&M Marses of the world, et cetera, in talking about how we can collaborate even in those countries with our activities, whether it's connecting our entrepreneurs that we work with their incubation and innovation funds that can get them to grow local businesses and move things along. They have a vested interest in making sure there's social and economic development in these parts of the world too. And if you have a reputation to be able to pick up the phone and walk into those large companies and NGOs and others, because you've developed good collaborations with them through other means. It really just advances the overall mission. So I think, to me, those are the really important factors of why you have to engage across the board geographically, but also in the dimension of level. You have to create an institutional program.
Let me ask you, follow up on that question. You know, one thing I do when I go out and promote our region everywhere else is I talk about how easy it is to do business and work with the universities and collaborate. And I always use the example of BASF because we've got great stuff online about the partnership between NC State and BASF. What can we do to engage more big business globally? How do we get our name out there better? If that's one of the things you're saying we need to do.
And I think across the university, we probably already do that individually as faculty. At CALS, I know that, we have well-known faculty. Craig's a great example of that. He's not just well-known for work and development, just generally in the genetics and breeding and potato. I mean, the sweet potato is just known everywhere. So those are all, seeding strong, hiring smart, seeding strong young faculty, making sure that they're successful and letting them get to a point where they're ready to then take on development activities. This is why I say international efforts and recognition are different than engaging in development. And you need both. But what we should be doing, for example, is encouraging our faculty from an NC State perspective to be more engaged in international conferences, grant activities, collaborations, etc., setting up opportunities that open doors so you can shake hands, I guess, in a post-COVID world with people and actually get to know how people work, whether it's in Italy, for me, because I like going back to Italy, or in Germany, or in Japan, or in Australia. Those are all good things, right? And that's a good first step. But then when the opportunity comes to move into different dimensions of that international work, whether it's development or others, we need to find ways to seed those efforts so they can kind of grow those connections in a real way. And I think it goes back down to, I think we've discussed it before, this is truly an issue of institutional commitment to this. And Dean Lommel kind of said that this is something that NC State is prioritizing. And I think we will see we will see the fruit of that investment for sure in the years to come.
Jim, let me bring you back in. Obviously, from where you sit, you deal with a lot of different folks all over the world. Do you want to add to what Mario was saying on what you think NC State could do more to kind of build our global reputation?
Yeah, it's, from my perspective, largely a matter of engaging, and sure, some of the programs that we and others do bring opportunities because you're bringing people shoulder to shoulder with the big companies. I'm mostly in breeding and genetics, so I think of the large seed companies besides BASF. You've got Syngenta, which is an RTP as well. Corteva and Bayer are the other big ones. And these interactions that Craig now has with global leaders from those companies, it also brings a lot back in terms of methodology. And when you get these international fora, people from the big companies are coming together, and they can't talk the specifics, but in terms of generalities and basic principles, there's a ton that comes back to NC State. And even, I don't know if you're aware, you've hired Carlos Iglesias out of Syngenta, who brings a wealth of industry background and a wealth of plant breeding background to sort of bring those things back to NC State. And yeah, so engaging abroad, bringing people in, sending people out, the more you're interconnected, the more it brings opportunities. And as Mario mentioned, recruiting internationally, giving young faculty chances to grow, you're building your future. And I think, and enriching the student experience as well as creating future opportunities and collaborations.
Okay. Tomohisa, we had the question I was going to ask for you is obviously you've had a presence here for 35 years. Do you think it would be beneficial for NC State to create some type of office like yours in other locations globally? You're muted.
This might be a very difficult question. My answer is yes, but with some conditions, a few conditions. Only if NC State has a clear visions and strategies, and if NC State has can give a clear mission to the international office, and only if NC State can commit more than 10 years financially, because it takes time. And my just small advice is to NC State, if I can, is get some focus. I mean, getting the world reputation from all over the world in all the field is impossible, of course. So NC State need to focus some market and some areas. For example, Nagoya University is focusing the only, if we focus in the United States, is that we focus NC State universities.
I think we lost him.
Am I back?
You're back.
So again, we need to focus. So for example, Nagoya University is, it used to have maybe 30 strategically important universities globally. So I just advised the president, it's too many. So we focus on 5 most important universities all over the world. So in the United States, we chose NC State universities, and Shanghai, some universities, and Thailand, 1 university. So we have only 5 now. So that kind of approach might be helpful to NC State University. Sorry.
Paul, I know you and I talked about this. Do you want to add on what your thoughts are that NC State could do a better job of?
Yes, I agree on the focus. I think it's just a matter of defining those goals and defining what do you want to get from the international collaborations and the international presence. Either if it's fundraising, if it's knowledge generation, if it's talent attraction, or if it's all of it, then each goal needs to have an activities plan in order for us to be able to get there. I do think that that collaborating internationally towards solving global problems, or actually having the mindset of extending what will be the impact, not only in the United States, but abroad of this problem, might bring different results.
Okay. I've got one more question. I'd love to actually turn it over and let anybody else on the call. We've got this great panel. So if anybody has questions, get ready for those. Mario, I had one question for you from personal experience. Have you seen, like, if international students have come and studied at NC State, have you seen any examples of them going back to their home country and then building relationships back with NC State?
Kind of broke up. Can you say it again? Sorry, Ryan.
Sure. The question I was asking was, in your experience, have you seen any success with students that come here from international locations, study, go back home, and then turn around and build relationships back with the university?
Oh, absolutely. I think not just my group is what I would say on the younger end of that. So most of the PhD students I've had have gone back and are now in director positions or faculty positions, whether they're government labs or academic institutions. But with my collaborators on multiple projects that I've had, whether they're from Cornell or Rutgers or here at Purdue, or even some that I've known through NC State days, actually, there are several examples of folks that when they get their PhD, through a project at Purdue, coming from Africa, let's say, they go back to their home country and they migrate up that chain. I think this has come up before. The next thing we know is when we have a call for another opportunity to develop a project, or we want to begin seeding a new project, we have built-in collaborators in the country already. So it's very quick to build our network. We just went through this in putting together a project proposal where within 2 days we had 7 countries aligned, because our former students are now running most of the national food science labs, or part of the CGIR or whatever, and we can begin to, what we would say now, leverage, right, our connections and our ability. But there's another aspect to that. They were fundamentally trained in our labs and our groups and our culture of research. So being able to set up that collaboration quickly in a timely fashion, and then show people that there's existing relationships to convince funding agencies to invest, or NGOs or others, that's important. Like, to show that you have that ability to put things operationally in the way they should be, to engage in that work. You know, we've done that often, and I would say a lot of times the projects and things of those natures bring back learnings that we have, you know, partners in companies in the US that become very, very interested in the type of research that we were doing in Africa on different types of fortification strategies, or use of sorghum and millet for gluten-free products, et cetera. So you have opportunities to really kind of, what I would say is really cultivate both sides of this. But that pipeline of students and postdocs and others is what enables and provides a sustainable, what I would say, foundation for these international programs. And that's in any country. You know, my collaborations in Italy are kind of born the same way. So maintaining a pipeline of students from those countries enriches our ability to be, and makes us more competitive for those opportunities.
Great. Well, let me open it up now and see if anybody else on the call has any questions they want to ask. Feel free to ask away.
So let me jump in. I don't have a question, but I want to build up in what Mara just said. That is totally true. So all this relationship, you will see how powerful those universities that you mentioned at the beginning, you know, the Cornells, the UC Davis, how they have the network spread around, and how they build so easy this huge projects, because they have people on the ground. They are their alumni. But it's not only that. I mean, all these students that comes here to the US to study are the brightest of their country. So they are highly selected. They come here, they get their education, they go back, and the connection is to the university where they study and the state. And if they become the ministries and they become the future leaders of the country, any business they do, they will do it directly with that connection. So that is a powerful network that only builds and only can pay off in the future. I only want to see the additional part of the business side and the political side of that type of relationships.
Great. Thanks, Jose. Any questions for our panelists?
Hey, Ryan, this is Alan Ayers. I got a quick comment and a question.
Okay.
And maybe it addresses what the panelists talked about this morning. By the way, I'm very impressed with the discussions that's been held so far on the panel. I'm Alan Ayers. I used to work with Bayer Crop Science, retired after 34 plus years, and now I work with the Innovative Vector Control Consortium on controlling malaria. And so it's already been mentioned. I think it was by Jim and maybe by others. There's a lot of opportunities to address challenges which agriculture directly impacts in areas like Africa and other areas. But there's other concomitant issues ongoing which indirectly impacts agriculture. So in IVCC, we're working with companies on use of insecticides to control vectors. And of course, this is malaria and chikungunya and Zika and things of this sort. And these vectors impact human health, and so does agriculture impacts human health. So we have found out, and it's been shown through lots of studies, if people are not well, they don't produce agriculture very good, no matter what your technology is. You can have great breeding, you can have great input, but it still has an impact. So as a university, you know, we've talked many times, Ryan and Steve and others, about the Plant Science Initiative, how you build these collaborative efforts, which many people on this call, especially Bill and Melinda Gates, have built all these coalitions to work together to connect a lot of dots instead of just looking at producing one technology or one innovation. So I think that if NC State can work through PSI, the individual centers of excellence, the departments, the ongoing research, and then ask, you know, what are we doing here that not only impacts North Carolina, but the nation and globally? You see a lot of opportunities out there to bring more light to be shown on the university and its efforts. So that's kind of a comment, but what can we do to, you know, to reach out and connect a lot of these things in a more systematic approach? That's the question, or the challenge.
All right. Jim, do you have any thoughts on that?
Yeah, great point. And you know, it is through collaborations and realizing connections and spin-offs, you know, or something that comes up in vector control, you know, has impact across many different fields. So thanks. Oh, Alan, nice to see you, and good to hear from you. So by building connections, by deepening relationships, that's where the future of innovation is going to come from, and where you realize what's possible. What's, you know, as more and more every discipline realizes the need to be multidisciplinary and to draw from many different strengths, you know, big data and different types of machine learning. It comes across the board. You find it's impacting whatever field you're interested in, and the big companies are realizing that, you know, where they used to have just a few defined disciplines, they really need to go much broader and have diverse teams, because that's where they're going to get the best results, the best innovation, by seeing things from different perspectives. You realize new possibilities and how to make your own work better and more efficient, and how to then build the networks that we heard from Mario, that you get succeeding generations of students and leaders that you're connected to, who then bring in new opportunities and new connections. It's, you know, Ryan, you're in just a great position to help NC State grow in that area of building their brand, building their connections, encouraging faculty to reach broadly, and that's where they're going to get the ideas, the inspirations, the possibilities for tomorrow's pressing problems.
I just wanted to add a couple things that I think made me think. Your question, Alan, was really good. It really made me think about, hey, how did I get into this? And why did I get into this? And I think what ultimately, it goes back to maybe, Ryan, what we talked about before, being present. Everything changes when you're on the ground. And we know on a macro scale what's important, but everything changes when you see it locally. And whether that's in Africa or in rural North Carolina, problems are local. And what I've always been amazed by in terms of like interconnecting things is how I wear a very different hat when I go on these development projects, and I'm no longer the phytochemical, secondary metabolite, bioavailability, pharmacokinetics, pharmacotoxicity person. I'm a food scientist. And oh, by the way, I can help people in terms of putting together a processing dossier or paperwork to file a process to make the product safe and everything else. And we know a little bit about fortification, and we know a little bit about that. And we need to do a cost analysis. but you start piecing things together in a very local, to address the local issue. And so the multidisciplinary thing that's impressive, and why universities like NC State are in such great position to address these complex issues of human health, even though we don't have a medical school, is that we can actually address very practical solutions by drawing on the talent that we already have. We are such a broad university that we can absolutely bring in expertise that we as individuals don't have. And it's such a great environment to be in from that perspective, and a strength for when we work locally. But I go back to that first statement. The problems are local. So you really have to be on the ground to see, to identify. And for example, on human health, for us, we were always focusing on commercializing products for sale. And then we realized there were opportunities in developing similar products at lower cost in rural areas to actually replace corn soy blend, which is the food aid product that comes from UNICEF. And why does it need to be replaced? No one bothered to do sensory work and see if kids even wanted to eat some of these products, but they'll eat the locally produced products. So we're solving a very simple health problem, or a big health problem with a very simple solution of making things locally with local flavor, with local ingredients, that we're able to build a small economy. I never thought I would be doing that type of work, and it's not what I do in my pure research thing, but it's this ability for NC State or Purdue, or, and where I was at, to bring in people to actually take and do the practical things. And I think that has implications on health. Like you said, even though we're not necessarily doing large intervention studies on this, it has implications on health, and it can be kind of amplified. So I really liked your point about how do we address the bigger challenges. And I think it always starts locally, though.
That's exactly where I was coming from. And in today's technology with analytics and things, we can crunch that data. We don't have to sit around and say, what if, I wish I knew. And, we have people in teams, and economists, and in the internet, cyber world, the whole thing, that can collaborate on this. You can see what a sweet potato production in North Carolina, what impact it could have in South America, Africa, everywhere. And that's something we can do right now. So the Plant Science Initiative is set up to do that. So that's why I'm such a big proponent of it. And hopefully my name on one of the bricks in the building over there is kind of small, but it's my support.
We appreciate that. We really do.
And it's so good to work with a group like IVCC that's really playing in a role as a PDP. It's a product development partnership, working with industry, academia, governments, the whole thing. And so that collaborative effort, we have several models on this call right now. Jim and Bill and Melinda Gates Foundation is just one of them. There's just several. And all we have to do is just tap into those and see what works.
Yeah.
And capitalize on it.
I would like to echo that by highlighting that the Plant Science Initiative is a great platform exactly to promote collaboration. I am convinced that formal collaboration comes from the informal collaboration, and the university and the biotech center and the different government organisms that are in charge of establishing those opportunities of collaboration are kind of the place to start, because by deliberately opening up those spaces where people come and freely share what their problems, their ideas, that is the start of all. So I really like the fact that the Plant Science Initiative is kind of the place and the foundation where all those informal collaborations and the informal collaborations will take place.
I totally agree, Paula. I think, working in industry all the many years, we always wanted to work with universities like NC State and UC Davis and Texas A&M, and because they provide a safe place. They do good research, they got good reputations, and it's not what a Bayer or a Ventus or all my predecessor companies said. we did this work in collaborative efforts. So it brings a lot of credibility to the whole story.
Let's just check and see if anybody else has any other questions for our panelists before I turn it back over to Adrienne.
Hi, my name is Alana, and I do have a question for all the panelists. At different points throughout the conversation, you mentioned the importance of being present. And within that component of being present, can you talk about the importance of listening? Because that, at least in my experience in doing some international work, that's where some of the challenges in communication fall, is in the listening component, because we have an idea of what the solution is, right? But we don't have that experience of being in that particular environment. So can a few of you just talk about that and how that can be improved?
I mean, I'm happy to start. I kind of build off what I said before. Being present is important because you can observe what the real problem is. When we're often asked, hey, we want to develop, new food fortification strategies for this part of the world or whatever, you can go to a textbook, pick them out, and make a recommendation. But if you don't really understand what the problem is, which implies that you need a lot of not just listening, but observation, it's important to actually physically be on the ground to fully characterize the challenge that you're dealing with, then what I would say are the strategies for solution. The other thing is that we noticed in the listening part of it, obviously such cultural differences in terms of how meetings start and everything else, and how things are organized. So when we have our, we'll have a training session in Africa, and the first 4 hours are dedicated to whatever government officials have to come in and give their speech. And we have to allocate an extra half a day just to be able to do this appropriately, so then people can actually have a good exchange of ideas. The other lesson I've learned is never, do your best to not go in with a preconceived notion, because trust me, things will be different on the ground. And we have tacted so many different ways on our project and adapted to the situations at hand on our development work, that we feel that we're just living it every day, if you know what I mean. And part of that is again, developing the relationships with the collaborators on that other side, whether they've been your graduate students or postdocs, or you were collaborators before. That trust that gets built over that time allows you to be very honest, but at the same time, it actually facilitates your ability to listen and trust what you're listening to. Because otherwise we're just not trained. Our brains don't function that way. My wife tells me I don't listen, so I know. But at the same time, we realize how much better things go when we move to that part of our brain where we shut off our mouth and we open up our ears.
Yes, I agree with that. And based on my small experiences, just 12 years, I have been working to connect dots and dots in Japan and the United States, and at the NC State University and other universities. Listening is the most important. Listening to the client's needs and try to find something common, and listening the patiently and without any prejudice and open-minded. I try to listen potential researchers who can collaborate, and just distribute those informations and listen and communications. Because of the 13-hour time difference, as the communication is very difficult and the listen is very difficult. But I have been working in that way so far.
Okay, any other final questions? Well, thank you to each of our panelists. I know Jim dropped off, but to the remaining panelists, thank you so much for your time and for the great discussion. And I will turn it back over to Adrienne.
Thank you for the invitation.
Thank you.
Thank you, Ryan, and thank you to all of our panelists. That was really insightful, and it's great to hear you all have a very diverse background and come from different parts of the industry and academia, so it's good to hear everyone's different perspectives. We will advance on to our faculty research presentations. So we'll have our faculty do their presentations, and then we'll have some time after that for questions. You can either submit the questions through the chat, or you can do as we did now and unmute yourself and ask the questions. So first, I will hand it over to Alejandra Huerta, who can lead off our faculty presentations.
Thank you, Adrienne. I will share my screen. And first of all, can I just get a nod to make sure that you're seeing the presentation mode? Okay, so I will, hi, my name is Alejandra Huerta. I'm an assistant professor in the Department of Entomology and Plant Pathology here in CALS at NC State. And I will be talking to you or giving you an introduction really about antimicrobial peptides used for, by phytopathogenic bacteria for interspecies competition. The pathosystem that I will be talking about, or the pathogen and the host that they attacks, is Ralstonia zoonocereans. Ralstonia zoonocereum really is a species complex of a lot of different bacteria that are genetically almost similar, but they vary to a great extent at the genetic level. They're the causal agent of bacterial wilt disease, and bacterial wilt makes the top 10 bacterial diseases limiting global agricultural productivity. It infects over 200 species, over 52 different dicot and monocot plants. Because of the wide host range and the large genetic distribution of the pathogen, it's really hard to manage and control. In potato alone, it's an estimated $1 billion losses a year. And when we see here the global distribution and these yellow dots throughout the world, we can see that a lot of the diseases found in tropical and subtropical regions, mostly inland and South America, Africa, and Southeast Asia, where we also see a lot of the food insecure populations throughout the world. Some of the most important crops are banana, tobacco, potato, peanuts, plantains, and tomato, just to list a few. But just, I can't stress enough the impact it has on a lot of different vegetables and important crops. So Ralstonia zoonocereum, because of its distribution, it's easy to work with in terms of international component. For my current project, I'm working with Dr. Jose Velasquez Jr. from Universidad de São Paulo in Brazil, Dr. Amilcar Sanchez from Universidad de San Carlos in Guatemala, and Dr. Jill Silvia from CIRAD at the University de La Reunión. And it's given me a really good opportunity to actually really study the diversity and really learn about the impact. And even though you see these yellow dots here in the US, the disease is really only located in the southeastern part of the United States, where we have more of that subtropical region and temperature, and where we also grow a lot of these really important crops that are susceptible to the pathogen. I have to give you an introduction to the life cycle of Ralstonia zoonocereum. So Ralstonia zoonocereum is a saprophytic bacteria. It survives in the soil for extended periods of time, and this is what makes it really difficult to manage. Its long survivability in the soil. It chemotaxes or moves to the roots of a susceptible host, where it enters via lateral root emergence or agronomic practices in the management. It moves from the roots to the xylem via the root cortex. There it starts to multiply, and it releases a lot of proteins that modulate the plant host for the bacterial survival. It colonizes the xylem and multiplies really quickly. And it's that multiplication in association with the production of this extracellular polysaccharide, which is this goo that you see here on the tomato stem. It blocks water transport from the roots to the shoots, leading to bacterial wilt, which is the name of the disease. And here you see a picture of the bacterial cells quickly multiplying and really blocking that xylem vessel in the tomato stem. So now we're going to get a little bit into the data. So the large distribution of the bacterium can also slightly contribute to its classification into different clades, more or less. You have a clade that is part of the endemic population in North America, a different clade that's endemic to South America, one of Asian origin, another one of Indonesian origin and African origin. So what we've done is that we took these different strains, in particular GMI 1000, K60, and UW 551 that are representative of bacterial strains from these different regions that have adapted to the particular environmental conditions in these regions. And North America is a warm temperate condition, and South America, the highlands in particular, is a cold temperate. And in Asia, it's more of a tropical warm temperate. So we put these different strains to compete, and we found that K60, the southeastern part strain, was a really strong competitor in tomato rhizospheres and in tomato stems. And this is shown by this pathogen population heat scale, where the darker the color, the higher the bacterial population, and the lighter the color, the lower bacterial population. And then the higher the triangle, that's where the majority of the population stands for. So for example, GMI 1000 had a lot higher bacterial populations when inoculated with UW 551, K60 against GMI 1000, and K60 against UW 551. And when we looked at the tomato stems, we also saw something very similar. When we then looked at in vitro studies, we also found that K60 was inhibiting the growth of these other two bacterial strains of different geographic origin. So just to give you a background here, the hazy background that you see is the target strain, which in this case is GMI 1000. And then these little holes in the agar is where we put the producing strain, in this case, either in green GMI 1000, in red K60, or UW 551 in blue. So when we have GMI 1000 as the target strain, we see that GMI 1000 did not inhibit itself, K60 did inhibit GMI 1000, and UW 551 did not inhibit itself. When we had K60 as the background, which was the strongest competitor in the soil and the rhizosphere, we also found that GMI inhibited K60, K60 did not inhibit itself, and UW 551 did not inhibit itself either. And then the results when we had UW 551 as the target strains were very similar, where both GMI 1000 and K60 outcompeted UW 551. So these in vitro studies correlated to our in-planted studies indicated that K60 was a really strong competitor, and there was actually in vitro strain-to-strain inhibition between these different strains within the species. When we looked at a wider population, which in this case was about 3-4 years ago, where only 27 different strains of Ralstonia were publicly available, and that we did one-to-one inhibition studies. Here on the left, you see the 27 different strains that are the producing strains, so the ones that go in the hole. And then in the top, you have the target strains, so the background strains that are inhibited. So we see that Granada 9.1, for example, does not inhibit Granada 9.1, but Granada 9.1 does inhibit to a small extent CFP P6438. And here in the bottom, you have that scale of inhibition. What we were able to find is that at least in our preferred strain K60, which is of North American origin, it inhibited the growth of about half the strains we tested, and it was also inhibited in return by about half of the strains. When we looked at GMI 1000, the pattern was very similar to K60, but when we looked at UW 551, which is the poor competitor against these other two isolates, we found that UW 551 did not inhibit any of the producing strains and was inhibited in return by about 60% of the strains we tested. So within these, within this screen, we only had access to 27 different strains, but in the last 4 years, and in collaboration with Gilles and Amilcar and Jose, we've been collecting different isolates from different parts of the world to have a better representation of the inhibition spectra of these strains, and also study the antimicrobial proteins that are actually determining this inhibition. So this is actually a gene cluster. That's what produces this antimicrobial peptides. It's a toxin-antitoxin system, and what that means is that you have a toxin protein, and directly downstream of that, you have an immunity protein that inhibits that protects the producing self of self-inhibition. Why would you produce something that would kill yourself, right? So just to give you a general background of what the toxin looks like, you have this toxin, you have the N-terminus of the protein, which is about the first 3/4 of the protein, and that carries the N-terminal region, which is involved in secretion. So this protein has to be secreted out into the environment in order for it to be effective. And then this gray component is the CT terminal, or the hypervariable tip. And this hypervariable tip is actually what carries the killing domain, which targets the RNA, DNA, or cell structure of a target strain. And then, like I said, directly downstream, you have the immunity protein. And when we look at this whole inhibition spectra, and we look at the diversity or the predicted number of strains that are available, predicted number of proteins that are available and that are present in each strain, we found that it varies. And through machine learning and also improved genomic sequencing abilities, we've been able to really narrow down the number of predicted toxins per strain. Where before we thought that Granada 9.1, for example, have 17, we now know that they have 4. However, when we look at these numbers, once again, K60, which is that one population here in the United States, it still has the largest number of producing toxins within the strain, and we don't really understand why. So with the help of my collaborators, we are hoping to take a machine learning and a comparative genomic study to look at all of these produced peptides and try to use that in order to generate some sort of management strategies for this soil-borne bacterial pathogen that is very difficult to manage. So with that, I'll take any questions.
Does anyone have any questions for me? Let's see. Nothing from the chat.
So just if there's no, I know that one of the things that in talking about international agriculture and possibly, how can we benefit? The world distribution of this bacteria is pretty amazing, and the diversity within it is great. And it'd be great if this is a problem in your country or any of the hosts that you're working with. I'm more than happy to work with you to try to develop some sort of collaborations within this particular area.
Thank you, Alejandra. And we will turn it now over to Raymond Gutierrez. You can share your screen.
Okay, do you have that?
Yes, we can see that. Thank you.
Okay, great. Okay, well, thanks very much for inviting me, and thanks to all the participants. This will be a slightly different style than Alejandra's very nice presentation, but I hope they both work. I'll talk a bit more broadly about my research and sort of where it sits, and most of my research is about technology and behavior in economic development. Can I advance? So, if you remember one thing from this talk, I hope you'll remember that behavior matters. And at a sort of engineering and ag science centered school, we believe very strongly that improving technology is critical, and it is, but it's not enough. Behavior still matters, and I was just thinking about an example. When I think of technology, I'm thinking fairly broadly. So this is a technology, right? This is a piece of cloth, and we know that this technology is extraordinarily powerful at protecting ourselves and protecting others from the recent unpleasantness, as we like to call it in this household, or corona mess, as my daughter calls it. So this exists. It's very cheap. It works very well. but we need people to use it, and many people do, and that's great. But adoption is maybe not as high as we would hope, given this technology's enormous benefits to society. And economics has a few things to say about why that might be the case, but lots of social and behavioral sciences do too. So this is one technology that I think is fine. Here's a different version of the same technology, right? That I find myself much more prone to use. It's got a great logo. Hope everyone can see. It's basically the same. But in order to get myself to use it, I've had to make a few modifications, and I really wish the good folks at the athletics office or whoever came up with this had thought at the time. You've got to put.
Raymond, we can't see the logo.
Oh, can you see the logo now?
No.
Oh, I'm sorry. How do I put the camera back on me?
You mean, oh, you're showing it through your screen? Okay,
yeah,
I see what you're saying. Okay.
I hadn't planned this.
No, you're fine. I was just looking.
Yeah, so if you go to my little there. So I had to put on this little nose piece. I had to find these, and I didn't know what these were called, which was made it quite difficult. Ear loop adjuster thingies was not a great Google search, but that sort of adding some features and then adding the appeal of having our wonderful institution's logo made it much more enjoyable to use, and then this made it a little bit more comfortable. This thing made it stay on my face. So this is just kind of a not so silly example of the fact that the exact features of technologies need to be very carefully considered in order to get people to use them, and to use them consistently and use them well. And that's really what my research is about. So, many of you are real scientists, unlike me, although economics is trying to improve our scientific rigor. So you've I'm sure you or your institutions have developed what would seem like a very beneficial technology. And again, thinking very broadly. So I mentioned of what technology means, just a new way to achieve some goal or produce some outcome. And that's wonderful, but it's not automatic that households or firms or farmers or whatever the intended sort of use group is are going to adopt it. And so what we do is work with all kinds of social scientists to try to figure out what some barriers to adoption might be. And some of these are sort of general from kind of behavioral science. Some of them will be very case specific, like the little nose piece, right? And what interventions can we make to help overcome these barriers to adoption and consistent use? And then finally, there are lots of cases, things like weatherization of homes for energy savings in this country, or the use of clean burning cook stoves in many countries around the world, where a technology exists and seems to be used somewhat consistently, but doesn't provide the benefits that we might have expected just from sort of an engineering approach. And so we need to evaluate kind of why a technology produces the effects that we had hoped, or maybe falls short, and how we might try to improve things. So how can we learn what works and why? How can we learn how to integrate insight from behavioral science with technology? Well, we desperately need to use rigorous evaluation methods, and there are many of those. My kind of preferred flavor, when I have the money, is to run a randomized controlled trial. So how basically how this works is working with practitioners, with the intended users, and with other social scientists or behavioral scientists, we develop a theory of how technology and behavior interact and what the sort of main barriers to adoption or effective use might be. We design interventions to try to sort of address or overcome these barriers, and then we implement those and try to test their effects in a rigorous way. For example, a randomized control trial with a household. And this, when I say barriers to adoption, I mean things like price, things like convenience of use, certain attributes that may be appealing to a household that we hadn't anticipated ex ante. So just a couple of examples of projects that I'm trying to put together in different stages of development, just to kind of give you a flavor. So in many parts of rural sub-Saharan Africa, as many as half of local water points, and these are typically manual hand pumps, are out of service. And that's obviously a real shame, because while it's a very simple technology, it can provide, it can sort of relatively inexpensively provide large quantities of high quality groundwater for human consumption. And so what many others, certainly not us, but a long history of finding that getting a community to save for basic maintenance and repair is often quite a challenge. And because of the failure to collect, say, $50 over the course of a year for routine maintenance, or the inability to contact a skilled repair worker, a hand pump and bore well that costs several $1,000s to install and is providing a fantastic service to that community can fall out of service. And so we've been trying with Water Trust Uganda, a Ugandan NGO. They have developed this really nice sort of intervention trying to promote local institutions that can effectively manage the water point. So that involves a lot of things, but primarily involves regularly saving for routine maintenance and for either simple or emergency repairs. And so we've been so far unsuccessful with a few grant applications, but we're still learning and still working, trying to get money to run a randomized controlled trial of this intervention that they have implemented quite successfully in kind of a descriptive way, but without a kind of rigorous and large scale evaluation. And with some colleagues, Zach Brown and Rod Rejesus in my department, second example of a potential project, we've had some very preliminary discussions with folks at the International Rice Research Institute, thinking about some of the new varieties of rice and practices for paddy management that they've developed that can increase resilience to basically to climate change, to droughts, and to floods. These are wonderful technologies. It can be difficult to get farmers to adopt them, and so this is quite preliminary. We've spoken to a couple of people at IRRI about some ideas they have, and putting together sort of a pilot proposal with a view towards getting a grant towards a large scale evaluation of some of these strategies in a randomized controlled trial. So that's what I prepared. I'd love to hear from anyone about ideas you might have for evaluating interventions that you or your institution has created, or think a bit about sort of challenges that you faced in technology adoption, and how sort of social science can complement technological innovation. Okay, thanks.
Thank you, Raymond. I don't see anything in the chat right now. Does anyone have any questions? Feel free to unmute yourself and ask away. Right. I think we have got lots of information. People are digesting it right now. If you have any questions, just feel free, as you're going along, you can either direct them directly to our presenters or ask them in the chat.
Okay. Stop my screen share.
Okay, I'm hearing no one, so we will continue on. Our next faculty is Carrie Pickworth. Carrie, if you want to share your screen.
Good morning, everyone. I've been greatly enjoying the conversation and the discussions that we've been having, and I'm going to kind of take us outside of the box compared to some of the other presentations. And so my focus area is in academics, and so my main research and stuff is related to academics and academic programs. In addition, I am a ruminant nutritionist, and so I also engage in different projects related to beef cattle and sheep production and management and nutrition. And so I wanted to visit with you all today about expanding the horizons in terms of academic outreach. And one of the academic outreach programs that I started a few years ago here at NC State. And so just thinking, why do we need to have academic outreach? And just thinking, we've mentioned how much importance there is in presence in our interaction and building a reputation. And right now, in some recent studies, it's showing that in the United States, we have over 22,000 jobs in agriculture going unfulfilled. As we continue to expand the human population and we diversify across the country and globally, there's an increased need for sustainable food production. And these are going to continue to drive the number of jobs, both in the production setting and in research, technology, and development related to not only our animal industry, our plant industries, but also our animal industries. And so we need to continue to build our workforce development, and student education is where we really need to be expanding. Because even myself, as I think about it, as I grew up in a small rural community, I didn't know anywhere, didn't ever really think outside of the box of my land-grant university to pursue my education. And it wasn't until I got involved in my graduate education, particularly my Ph.D., when I started networking with international students and others in our graduate program, that I really started to invest and explore how important it is and how critical even what my small family farm was doing was impacting our state, our region, and in the global. As we think about animal science, here at NC State, we have about 825 students in animal science. But much like everywhere else in the country, over 85% of our students are intending or exploring the idea of becoming a companion animal veterinarian or an exotic animal veterinarian. The reality is less than 10% of these students are actually getting into veterinary medicine and ending up in careers in that field. So that leaves a lot of students needing very late term to explore other career options, or even post-graduation exploring career options. And also, as we think about our land-grant universities and our animal science programs, we are very limited in terms of diversity. Most of our student population is coming from white Americans. And so we need to continue to explore, how do we educate and how do we inform students not only about opportunities locally, but engage other populations and realize how much we can connect with others. So a few years ago, I started a pre-college program entitled Livestock Science Camp that was designed to try to recruit youth from various different sectors, both inside the traditional agricultural community and outside, to help them explore careers and connections in animals and animal science, and particularly our production livestock species, in exploring career opportunities and understanding the science that goes into animal production. So for the first 3 years of the camp, I was able to do an in-person residential camp, and we set it up where students moved into campus on Sunday, and they were living on campus throughout the week. And we have counselors that are NC State students that serve as their supervisors and key in their education. Due to the pandemic, we've had to change that format. And so last year, and it's looking like this year, we're going to be doing a virtual program. And with that, we've actually had a lot more challenge. I thought it was going to be a great opportunity to expand our net for student recruitment into the camp program, but unfortunately, it's not opening up the doors. As we look at what kind of students we've had participate, our student demographics have matched very similar to our animal science student demographics, being predominantly female and predominantly white students. We have been able to recruit some students out of state, and we did have a few students come internationally to participate in the program. One of the biggest hurdles with that is that since we are engaged in our farm animal units and educational hands-on experiences on campus, it does require that we meet biosecurity guidelines of a 2-week quarantine before students from international programs could come onto the farms. We've also been able to enhance, and one of our target areas has been diversity and recruitment of low-income and first-generation college-seeking students. And that's an area that I would love to continue to build on, because we realize that we need to have students coming into our programs that didn't come from production agriculture and haven't thought about how much science, technology, and advancements there are in our global food security coming from connections with our agricultural industries and our animal industries. One thing that I do throughout the camp program is evaluate students and have them do a pre-post evaluation, having them express their knowledge base of animal industries and experience with different animal industries before they come into the camp program, and monitor their growth through the camp program. And every year we can see that we've had very positive growth rates. You know, generally it's about a 20% increase in awareness and experience coming through the camp program. And these are even, we do this on a 5-point scale, and a student might come in at believing they're a 3 out of 5 and leaving going up to a 4 or a 5. And so we do see growth in even our students coming from farm backgrounds, that they're getting a lot out of this and realizing there's a lot more to animal science and to agriculture than what initially meets the eye. The other thing that I really find valuable with this program is listening to students that do not come from agricultural background and their preconceived notions. You know, 2 years ago was when we had a lot of lawsuits going in against the hog industry. I had numerous campers come in and say, I am 100% dead set against the hog industry in North Carolina. I want it shut down. They're abusing animals. They're killing our environment, and they're making the citizens of North Carolina sick. Through the camp program and exposure to not only producers, but being on the farm, engaging in the environment, they realized what they saw in the news was different. And then they wanted to become advocates for the industry. They wanted to get into the science and learn more, because they realized it was a lot better than what they thought, but they realized there was still opportunity to expand this. And so it's tremendous when we can capture these students through experiential learning and get them wanting to get engaged in building the future for our industry. And so just wanted to share some highlights and some pictures. You can see here These are just a few snapshots of the different animal species that we engage the students in. They spend numerous hours hands-on with the animals, learning about them, learning from not only our students here at NC State, but also our faculty and staff, and expanding their knowledge and interaction. And I think there's opportunities to expand this even further. And that's where I'm excited to engage with you all today is how can we continue to build these awareness, build educational programs, love to engage more as we continue to utilize technology more, to have more perspectives in virtual presentations and do virtual tours. If we can't get everybody out to your site, have people connect and speak and share opportunities in the industry. We also need continued financial support in which, because this is a residential camp program, it is a program that is not currently supported. It is not part of the USDA program like the Ag Science Camp is, or it's not supported through the National Poultry Association like the Poultry Science Institute is. There is a financial burden for campers. And so I'm continuously seeking opportunities to create more scholarship so that it doesn't matter what your economic status is, it doesn't matter where you came from, that this is an opportunity that you can explore and really open up new doors for you. I would also love to look at opportunities to how can we engage more internationally? How can we engage more nationally and internationally? Can we create some virtual experiences? Can we create an opportunity with a study abroad type program in which we can do an exchange, potentially taking these youth and really exposing them to what all is out there. And so just starting them through the door at NC State and realizing how global our environment is. So with that, I thank you for the conversations that we're having. I look forward to continuing to engage and expand my horizons and expand our horizons of our students through expanding our outreach.
Thank you, Carrie. I am seeing a theme amongst everyone that the presence and the experiential opportunities are, with students and any type of international work. And we're seeing, of course, during COVID time, that's being limited. But we're hopeful to get back to having in-person. So at least we're making use of this platform right now, and we can share this information globally. So, but it is, I think we're all yearning for those in-person experiences more and more. Does anyone have any questions for Carrie? I don't see any in the chat right now.
I do have a question for Carrie. What are your recruitment strategies for broadening participation?
That's an area that I'm looking at building on. Right now, a lot of our recruitment is occurring through connecting with students, reaching out to school programs, reaching out to guidance counselors. In general, one of our greatest recruitment tools has simply been the NC State website and pre-college programs office and their ability to share. We have a lot of students that come looking at NC State's page for opportunities, and that's exciting. The international students, they came looking to NC State, they came looking, and so, it does speak to the global reputation that we do have, that they came looking for the opportunity. But I would love to do more at being able to proactively reach out to other groups.
There is one question from the chat asking, do you have any ways that you are targeting minority groups?
So one of the ways that we've been targeting them is through using peer-to-peer mentoring and using our students that are more diverse counselors and having them reach out to their communities and connecting with them. We have been working with the Office of Diversity, Inclusion, and Equity to try to help recruit. And we do have a scholarship program that is specifically targeted towards minority group students and making sure that that financial burden of the cost of the camp program is not a limiting factor for them by having those scholarships available to them.
Thank you. Thank you, Carrie. Appreciate that. We'll move on to our next faculty presenter, Trino Sancho-Ibanez. Trino, have the floor if you want to share your screen.
Thank you. Let me attempt that, and then we'll be on our way. So, can you see my screen?
Yes.
Thank you very much. So first of all, thank you for the invitation. It has been a very, very interesting morning. I have learned a lot, and I already make notes. I probably will be reaching out to some of the people that were present here, either presenters or panelists. So my presentation today is something that has just come up. We are still developing the tool that I'm going to be presenting, so it's not finalized, but it's a tool that has, well, we think it has a lot of applications in many different fields. And this particular presentation is with early biotic stress detection by a multispectral and machine learning approaches. And I will present briefly the problem that we are looking at. It's not the only problem that this device can be used for, but is the one that we are using it for. And there is a virus in Africa that is called the cassava brown streak virus. And it's a virus that renders the tubers of the cassava plant useless. One of the problems with that is that you can't see what is the damage in the plant. So cassava brown streak virus takes very long time to detect, and it requires difficult methodologies to take in the field. They are not impossible, but they are expensive and require expertise. The symptoms many times are just looked visually, and you need an expert eye, but even with an expert eye, they are confusing. And the main problem that we are trying to support with is that the propagation systems for cassava is propagated by stalks. You cut pieces of the plant and then you propagate the plant. It will, if it has the virus, it will propagate the virus, but you won't know that it has the virus until it's too late. Once the plants are very old, as you can see in the image on the left, you cannot see symptoms over to about 50 days post-inoculation in artificial inoculations. And you need sometimes molecular tools to actually know that the virus is present. So this is a challenge that is very difficult for the people doing the propagation, because this is a subsistent crop. And the people that buys those propagated plants, that then they want to plant them in their fields, and then later on, they're going to have their tubers not useful for commercial or not even for human consumption. So it's a big problem in that sense. Now, this solution that we have been envisioning, it's based on multispectral imaging of the cassava brown streak disease. So what we are doing is, first of all, we got our hands into an infectious clone. We didn't develop the infectious clone, but we asked for it and we are using it. So we have a controlled environment where we can inoculate cassava and follow the development of the disease. And what we came up is this collaboration with the University of Manchester, where an engineer there has developed a handheld multispectral scanner. So we apply the scanner that is represented there, and it looks probably like one of the Ghostbusters ghost capture devices. It's bulky and a little cumbersome to use. That contains about 14 different wavelengths of LED devices that are used to look at the reflectance of the leaves that are placed in the presence of that device. But after that, the information that is captured, this is where these collaborations are really extremely important, because I know very little about machine learning. So we have yet another collaborator at the University of Manchester that then processes all the data and then has been developing a machine learning approach where, with randomized capture of 12 or more patches on the leaves, avoiding the main leaves, the main veins, and the grid that the machine has, we have been able to identify much earlier than before the presence of this particular virus. One of the advantages of inoculating only with the virus is that we are going to be able to narrow down the signatures of this disease in the plants. And then what we are proposing after that is to use a multidimensional database that we are also collaborating with people here at NC State from the IBM company, to make sure that the processing of the data, because this is heavily, it requires a lot of computer power, that this can be actually be performed easily. And after this, once we get this going, we are gonna move to the field trials, and we have now collaborators in Tanzania, in the International Institute for Tropical Agriculture, that then we're gonna take the device to the test in the field, which is where we really want to see it, because there are many other stresses in the plant that might interfere with our approach. So we want to see if we can still capture the signatures that we have been found in the laboratory. So what can we accomplish with this device? Well, we can detect the disease early during the growing season. So people, not only the people that propagates the plants, but also whomever grows it, they will be able to detect if the virus is present early in the season, so they can replace those plants. But we are thinking that this will be more, the approach is better for people that does the propagation, because then they can cool all those plants that already have the virus. This is a low-cost device, it's not extremely expensive, it's easy to produce, and it can be placed on site. So you don't have to move a big machine, like when you do hyperspectral, sometimes you require a very complicated machine to move around. This is multispectral. You define which LEDs you put in your machine, and you get relatively fast results. You can have several hours, you will know if that plant is infected or not. But we didn't stop there. We are now producing the next generation of this device. We produce now a smaller, more compact and easier to use, and especially for thinking on the people that is going to be using in the field. The previous device, you needed to detach the leaf to make the scan. With this new device, the next generation, we call it, it's going to be, you don't need to detach the leaves, as you can see in the picture there. So the other advantage of this newer device is that we are also adding not only reflectance, but we're also adding transmittance here. So we'll have two different types of signal that will certainly enrich our ability and improve our ability to detect the virus at much earlier times. And we are focusing on viruses, but of course we can use this to solve other biotic or abiotic stress issues that can affect plants. And our idea here is to detect those changes in the plant much earlier than visually are, that you can detect with the naked eye, to make it much easier to have approaches to protect the plants or to prevent events that might be damaging to the crops. So I think with that, I hope I didn't exceed my time, and I'm open for questions if you have some.
Don't have any questions in the chat right now.
Question, Trina. So is there a threshold or a lower detection limit? And do you know, have you tried this with bacteria by any chance? Because with some bacteria, it's really, you only know right when you get disease, but it's easier to manage before bacterial populations get to a certain limit. So I was just wondering about that.
Yes, we don't know yet our limits of detection, and we are still fine-tuning the algorithms for the machine learning and how to scan and all those other options. However, we are open to try this with anything else. This can be applied to any plant, and as long as you have a way of inoculation, for instance, if you want to work with your bacteria, and let's say tomato, we're also working with tomato. So you can either render us the bacteria, or we can go to your lab and we can do the scans there, and we can find out if we are able to detect your particular bacteria earlier than you do with other methodologies. Of course, we can try that, certainly. We don't know, we haven't applied this to bacteria yet, but this is applied to other purposes. We're not inventing the wheel here, but we are applying it more carefully tuned to specific problems, like is the cassava brown streak virus. But Alejandra, that's a great opportunity to explore the abilities of this device. And I assembled this thing. It's 3D printed, so it's very easy to fix and repair and all those things. And it's a very useful device. I think that we should look for more applications. So we can discuss that later and see if we can help with your project particularly.
Hi, Trina. John Cruz with Plant Response. Thank you. Excellent talk. I've enjoyed it. Quick question about whether you're calibrating against potential nutrient deficiencies in the plant. And then how can you distinguish biotic versus abiotic stress?
That is actually part of our next approach. All right. First, we wanted to know because this was our main problem, this virus. So we calibrated for the virus. So next, and we put a bid for this. We don't know if we're going to get the resources because it will become more expensive. We're going to be growing this cassava under different conditions of abiotic stress. We will do some water stress. We will do some nutrient stress and light and temperature stresses. And then we're going to use the same calibration and see if that will interfere with our viral detection. And since we have the virus as well, we will do the same thing with and without the virus. And of course, we're going to start eliminating everything that is not. Other thing that we are doing as well, John, is that we are also changing the LEDs that we use. We found that 2 of the initial wavelengths that we use, they didn't provide any significant information. So we changed them. We removed those 2 LEDs and introduced 2 more that were closer to where we see the signatures. Therefore, now we are having even better results than before with the new device because we have been coming down to what we need. That's one of the advantages of this is that with the LED providing so many different wavelengths, you can fine tune to your specific need. So in the future, not yet, we are not done that yet, that calibration. In the future, we will certainly be able to know more if all those abiotic stresses are going to be affecting this or not, or if we can separate the signatures, which even if it affects, as long as we can separate the signatures, it shouldn't be an issue for detection in the field. Very good question, John. Thank you.
Thank you.
Any other questions? Just have a comment from Josh Dragel saying, fascinating device. Looks quite effective and promising in the comments. So it's very fascinating.
Well, thank you very much.
All right, not hearing any, we'll now turn it over to Josh Drabel for our final faculty presentation. The floor is yours.
Okay. Can everyone see my screen?
Yes, we can see it.
Great, thank you. Okay, I'd like to start by thanking the organizers for the opportunity to present in this year's Emerging Research Showcase. My name is Josh Drabel, and I'm a new faculty in the departments of Molecular and Structural Biochemistry and in the Plant and Microbial Biology department. My lab uses developmental genetics to understand plant architecture. So plant architecture is a critical component of the global crops that feed the world because the overall shape of the plant impacts how densely it and its neighbors can be grown in rows. Additionally, plant architecture impacts cultivation and also how effectively and efficiently grain can be machine harvested. So some of these crops have already been mentioned today. So millet and sorghum I know came up earlier. And I focus on maize. And so maize is, along with wheat, is a leader in total area harvested. For maize, when most people think about yield, they think about how many kernels can be packed on an ear of corn. And indeed, a majority of what we think about with yield does come from the cob. But yield is also driven by how many plants we can grow in a given area of land. And this is a really important concept because we need to be thinking about more effective ways to be growing more crops on less land, especially as drought and other stresses become more prominent. So shown nicely in this graph are data in the gains in grain yield over about the past 80 years. And so what we can see nicely here is that as plant density, shown here in these different indicators, as plant density has increased, grain yield has also increased in these hybrid maize trials. And so the increase in plant density is largely afforded by changes and alterations in plant architecture. So high density tolerant plants oftentimes, for example, have more upright leaf architecture. And this can be seen nicely in these row crops here that I've already mentioned. So there are several components to plant architecture, and my lab focuses on specifically a very selected area of the leaf. And so in the mature leaf in maize, we have the blade and the sheath, and that's separated by this boundary region here. Now this boundary region acts like a morphological hinge to deflect the blade away from the plant axis. So this can aid in more efficient light capture under high density plantings. And so over the years, we've had a lot of success doing mutant and genetic screens for variants that alter plant architecture. And shown here are just two examples of maize mutants relative to the wild type. One is called Ligulus 1. It has a very upright leaf architecture. And to contrast that, there's this mutant called drooping leaf that I discovered in my PhD and continue to work on today that has a more open canopy or more relaxed leaf architecture. And so the differences in these mutants here really comes down to the variation at this blade sheath boundary. So we know that in wild type, we have this nice articulated area here between the blade and the sheath, whereas in the Ligulus 1 mutant, that area has been deleted. So that causes a more upright leaf architecture. And in the drooping leaf, this is a more expanded morphological hinge, which contributes to the more open canopy. And so I'm a developmental biologist by training. And so to understand these macro phenotypes that we see in the adult shoot, we have to look quite early in development. And this is the key to my research, where we have a cell or tissue specific level of understanding the genes that are expressed. And then we use this to identify novel candidate genes. And so some of the approaches that my lab is using to discover new candidate genes that regulate leaf architecture. Well, we're taking a developmental genomics approach, and in one approach we've used in the past laser microdissection to precisely dissect out areas of the developing leaf, and then to isolate and sequence RNA from these developing tissue regions, and to identify hundreds of candidate genes that we then can use to test the function of these genes through mutation analysis, whether that be using transposons or CRISPR-Cas9. And then we find that some of these genes are very important for altering the structure of these boundaries, and they have major impacts in plant architecture. Another approach that we're using is single-cell RNA sequencing. And so with single-cell RNA sequencing, usually the shoot part of the plant has been very recalcitrant to isolating single cells. You have to go in and actually separate the cell from the cell wall. And so we've been able to do this now in maize, and we feel like this can be applied to other plants as well. So for this, we isolate single cells from the plant, and we individualize and barcode each of these cells so that we can determine the genes that are expressed in that given cell. And by doing this for thousands of cells, we can start to generate these cell clusters based on each cell's transcriptome. And then this becomes a very rich resource for mining for new regulators at a cell-specific level that we can go back and test to whether they alter plant architecture. And that's exactly where my lab and research program is at the moment. I just started my lab in January, and we have a good rich set of data that we're starting to either generate or mine these data sets for candidate genes, and then are performing developmental informed in-field genetic screens for new regulators of plant architecture. And so if you would like to know more about this research or opportunities for partnering, please be in touch. My contact information is here, and I'd be happy to take any questions.
I don't see any questions in the chat right now. So if anyone has questions, you can unmute yourself.
No problem. Yeah, and also they can feel free to email me as well if they come up.
Okay. There is a question in the chat. how much more room is there for an upright habit in modern corn lines?
I believe that, so one of the things that we've been more focused on recently is that at different levels of the plant canopy, throughout the life cycle of the plant, we can start to modify the architecture. So a lot of times when we think about hybrid maize, and we think about this upright leaf architecture, it's oftentimes leaves above the ear. So after the plant has transitioned to floral production, those are the upper canopy leaves that we think about as being very upright. But in the early juvenile phases of the development of the plant, those leaves are actually quite sprawling and they fill out the row more. So we haven't really started to modulate too much the different parts of the canopy throughout different stages of development, especially as plants go from a vegetative to a reproductive change in their growth. So I would say that the question's still pretty open as far as how much room there is. And I think it really depends on where in the life cycle we're targeting some of these modifications. I will mention that the single cell and the laser capture work that we've been doing has been really at the earlier stages of plant development, because those are the tissues that we can get at in large amounts. But I will say that some of those genes we're seeing an effect already in adult traits. Thanks for the question.
Any other questions?
Yes, I would like to ask Josh, how easy is that isolation of independent cells, and what's your loss versus what you can actually get with the next generation, the next step in your experimentation? It's just a question, but I'm curious.
Yeah, it's a great question, and it's a new, here at the Triangle, people have been trying to isolate single cells by either sorting cells by reporters. That's been something that's been really pioneered by the Benfey lab in the root for a number of years. But again, the shoot or the developing meristems in the growing tips of the plant above ground, whether that be from the inflorescence to the vegetative, those have been more tricky to actually get protoplasts from. And so, we always target about 10,000 protoplasts for each of our single run experiments, and we're using a 10x genomics platform. And so, getting the protoplasts actually is not a problem now. We've got a cocktail that helps us isolate healthy protoplasts from these meristem tissues. And we have no problem concentrating those protoplasts and giving the facility the amount that they require. So we are targeting around 10,000. And then after we run our sort of set of criteria for quality control, we end up ranging between 5,000 to 7,000 different, 5,000 to 7,000 high quality cells that we feel like we can then do genomics with. And so, there's a discussion in the field right now about how important replicates are in these experiments, because there's no such thing as a true replicate in this sense, because there's no guarantee you're ever going to recover the same cells. So we do try and do at least 3 replicates now, or we call them replicates, because it's nice to see that you'll have overlap for some cells, but then you'll also be gaining new cell types in other experiments as well. And the goal really is just to have as broad representation of the cells as possible.
All right, thank you. I want to express my deep appreciation for everyone who presented and contributed to the conversation, and of course everyone who attended, taking the time out of your day to discuss the important topic of international programs, especially in College of Agriculture and Life Sciences. We're saying it's critical to have international collaborations for successful science to happen. We will now be opening up breakout rooms for everyone to meet the faculty who just presented. I'll open those up and you can select the breakout room based on the PI there, and you can grab something to eat and have some lunch discussion with all of our faculty. So again, thank you everyone for participating, and look forward to another emerging research showcase come fall.
I would like to just mention real quick, if I may, that unfortunately I'll only be able to be in my breakout room